let's take a little gander into genes and life. All right, so our nucleic acids. In the previous chapter, at the very end, we looked at the fact that our nucleic acids contain genetic information. So when we look at our nucleic acids, we can get DNA, which is deoxyribonucleic acid, or RNA, which is just ribonucleic acid. Okay, so DNA, it's this huge polymer, thousands of these nucleides. We get two strands that come together in this double helix form, and this is where we get our genetic code for each individual. Our RNA is a single strand, so it's a single helix, not a double helix, and it contains ribose instead of deoxyribose, and we end up with a uracil base instead of a thiamine base. RNA guides the synthesis of our proteins in our cells, and so we get this process called transcription, and our DNA directs the synthesis of messenger RNA, transfer RNA, or ribosomal RNA. You may have learned about those in previous classes, but you might have seen like mRNA or tRNA or rRNA, which is how we abbreviate those. Okay. So DNA, we have cytosine, guanine, adenine, and thymine. In RNA, we have cytosine, guanine, adenine, and uracil instead of thiamine. Now, we discovered DNA structure through a lot of work. There's this technique called X-ray crystallography. And so we get these incoming X-rays, they diffract, and we get a spot. And so based on these spots, they were able to figure out the structure of DNA. X-ray crystallography is a really cool technique. I've done it. I did it for a while in research. And so, yeah, you get these like spot patterns, and then you have to, based on some other information, decode the spot patterns to figure out what you have where. Hey, and then this is what it looks like on the plate for those spot patterns that helps you decode that. Right? And so this is how we get the structure of a lot of our macromolecules, not just DNA, but a lot of our macromolecules, our proteins and stuff. We get their structure through this X-ray crystallography. And so the double helix, all right, we get from that spot pattern. So you can see there that hole coming in and out, that's where that double helix came from. Now, when we look at these base pairs, we see a lot of different percentages of base pairs depending on the species. So our species, we've got about 30% adenine and thiamine, and then around 20% guanine and cytosine. And other species, so fruit flies, have a more even mixture of them. And so you can see just different species have different ratios of these base pairs. Now, things base pair the way they do because of hydrogen bonding, that intermolecular force that we've talked about before. So we get these linear hydrogen bonds. So adenine and thiamine, they're going to form 2 linear hydrogen bonds, and cytosine and guanine are going to form 3 linear hydrogen bonds. Okay, more hydrogen bonds, stronger the tethering of stuff is, and if they're nice and linear and everything, they're nice and strong. So this is where we get our base pairing from. All right, when DNA replicates, we've got this DNA polymerase that comes in, and it starts working with the DNA to untwine or untwist the double helix. It's going to pull the things apart, and then the polymerase comes in and starts doing its thing. Okay, and so in our cells, we've got this genetic information. So we get our DNA, we do this transcription. So we have our messenger RNA and our ribosomal RNA and our transfer RNA that all come together to synthesize what we need. Okay, and then our proteins, they have different structures. There's a primary structure, which is the sequence of amino acids. Then we get this folding pattern from a secondary structure, and then this tertiary structure is this overall 3D structure. If a protein is miscoded, it doesn't fold correctly. If it doesn't fold correctly, we don't get the correct tertiary structure. Without the correct tertiary structure, it's not going to fit in the right spot, and it's not going to do its job. Some misfolded proteins are just, they don't do anything. They're dead. Other misfolded proteins can cause problems. Prions are misfolded proteins. Okay. When we talk about genes, the debate on genetic engineering comes up. So just in the back of your mind, in a face-to-face class, I actually run this as a debate. I have this whole point and counterpoint thing to think about with genetic engineering. All genetic engineering is doing is it's coding stuff specifically, okay? So insulin is produced in bacteria through genetic engineering. So if you know somebody that needs insulin, right? We can do a lot with our genetic engineering that helps us try and figure out, ways to make things better. Okay. One thing that they try to do with genetic engineering is to grow crops with different traits. So there's a big thing with pesticides, all right? We don't want to use tons of pesticides. They're not good for us. So sometimes what we can do is we can genetically engineer a plant to be resistant to disease or bugs or things like that. It doesn't mean that we are changing human DNA. All we did was change the DNA of a plant. We haven't done anything to alter our DNA. If we eat the plant, we are not altering our own DNA. We're not modifying anything about us. All we've done is we've made a plant that's resistant to diseases or resistant to pests where we didn't have to spray it with mass quantities of some sort of pesticide or antifungal agent. Okay? We can also make it so that crops, will produce more as well. And so there's a big, thing with GMOs. So for this class, I'm going to give you a discussion board to talk about GMOs for the point and the counterpoint for our final week together. So something to think about there, because the thing with GMOs is they sound really scary. Ooh, genetically modified organisms. But when you think about okay, what are they trying to do? Well, they're trying to make crops or other things that can resist stuff where we don't have to dump a bunch of, potentially really harmful chemicals on them. So maybe it's not such a bad idea after all. So just food for thought there.